Look at the two dark crescents sitting inside this larva. They are sacs of air, and every morning at dawn, the animal uses them to sink more than 200 meters into Lake Malawi, down into water that has no oxygen in it, where the fish that eat it cannot follow. 200 meters of water presses down at about 20 atmospheres. An air-filled space that deep should be crushed flat. And for most of a century, the fact that insects carry air inside their bodies has been the leading answer to a genuinely strange question. Insects own the land. More than 1 million described species in every forest, every desert, every pond and river. The open ocean has essentially none of them. In 1998, the physiologist Simon Madrill laid out why, in a paper titled plainly, "Why Are There No Insects in the Open Sea?" At the surface, in daylight, fish eat whatever they can see. Crustacean plankton survive that by dropping into deep, dark water all day and coming up to feed at night. Insects breathe through air-filled tubes, and air under pressure compresses. So an insect could not make that dive, and got eaten. That argument held for decades. So the obvious question about this larva is whether it means insects are about to move into the ocean. The answer is no. But the reason they are not has just become a much better question. Here is how Evan McKenzie and Philip Matthews found out what these sacs can take. They milled a pressure chamber out of a single disc of aluminum, about 11 centimeters across, bored through the middle to make a cavity of roughly 40 milliliters. 2 thick acrylic windows bolted onto the faces so you could see inside. A hose ran to a hand-operated hydraulic pump. That is the whole instrument. They flooded it with lake water, dropped larvae into little plastic cells so they could not swim off, put it on a stereo microscope, and started pumping. Pressure went up 30 pounds per square inch at a time, then held 3 minutes. The animals are transparent, so there was nothing to infer. When a sac gave way, you could see it collapse straight through the cuticle, and the larva would thrash for a few seconds. They kept going, well past anything the animal will ever meet. In one run, with a sac dissected out and pressurized on its own, the wall held to 819 pounds per square inch before it imploded. In fresh water, 577 meters down. Intact final stage larvae came in around half a kilometer. These animals dive 200 meters. The sacs are rated for more than twice that. But maybe insect airspaces were always tougher than anyone assumed, and nobody had checked. So the team tested relatives. The same 2 pairs of sacs are standard equipment across the whole genus. They collected larvae from Shirley Lake in British Columbia, a pond 8 meters deep with no fish in it and no reason to dive. Those sacs fail at around 10 meters. Lake Victoria species land in between. And within Lake Malawi's own larvae, crush depth climbs with every molt. None of that is standard insect anatomy. It is something this population built, under pressure from the fish above it. So what is holding? These sacs are not lungs. They are sealed off from the rest of the breathing system, and the larva takes no oxygen from them. It absorbs gas straight through its skin. Gas dissolves freely across the wall in both directions, so the pressure inside just tracks the water outside. There is no cushion of compressed air propping anything open. The wall carries the whole load by itself. And the wall is a composite. Under an electron microscope, it is visibly striped, alternating bands of stiff chitin cuticle and a rubbery protein called resilin, the same elastic protein that powers a flea's jump. The cuticle bands run around the sac like ribs. Squeeze it, and the sac gets shorter without getting any narrower. And the harder you squeeze, the stiffer it gets. That same construction is how the animal steers. And this is the strange part. Resilin is full of acidic amino acids. Put charge on them and they pull in water. Take the charge away and the water leaves. So the larva pumps protons into its own sac wall. The resilin gives up water and contracts. The sac shortens, the animal gets denser, and it sinks. Let the wall drift alkaline and the resilin charges up, draws water back in, and the sac lengthens again. It is a ballast tank run by pH, and as far as anyone knows, the only place in biology where resilin works as a chemically powered motor rather than a spring. And it runs in 2 gears. In the top 50 meters or so, the larva actively trims, working the wall's chemistry back and forth to hold its position. Deeper than that, it stops trimming and lets the sac ride. The stiffening does the work. The wall resists further compression, so the animal does not keep getting denser all the way down, and does not have to fight its way back up at nightfall. None of which would matter if the dives were only a lab result. So in August 2023, the team moored a sonar on the bed of Lake Malawi off Nkhata Bay, 275 meters down, pointing straight up, and left it for 13 days. It pinged at 4 frequencies because small, soft targets scatter high and low frequencies differently. That is how you tell a cloud of millimeter-scale larvae from a fish. And the record is a commute. At dawn, a dense layer peels off the shallows and walks down past 200 meters. It sits there all day. After dark, it comes back up. Separately, the team lowered a probe every 10 meters down to 250, measuring oxygen the whole way. In the northern lake, it runs out around 176 meters. Below that, nothing that breathes water can stay. Which is the point of the dive. There is nothing to eat down there. The larvae go because it is the one place in the lake with no fish in it. They arrive, drop their metabolism, switch to running without oxygen, and wait for dark. This is not a handful of animals. Counts run from a few hundred to over 12,000 larvae under every square meter of lake. And when they finish, they come off the water as adults in swarms so dense they look like columns of smoke on the horizon, thick enough that people along the shore catch them in baskets and press them into cakes. The paper came out in Science in July from Philip Matthews's lab in the zoology department at the University of British Columbia, with a commentary alongside it by John Harrison and Art Woods. Their verdict is blunt. The old hypothesis is refuted. An insect can carry air into deep water and keep it. Matthews's own line is that pressure is not the barrier we thought it was, which leaves the original question open and in better shape than it was. Insect blood is far more dilute than seawater, so a marine insect fights a steeper chemical battle than a crustacean does. Crustaceans got there first by a couple of 100 million years, and they still hold the niches. Matthew's phrase is that insects cannot move back into an empty apartment because the tenants are still there. The ocean is not literally insect-free. 5 species of sea skater live out on the open surface, hundreds of kilometers from land. But they colonize the boundary between air and water. The column below them is still empty. And Harrison and Woods add a narrower version of the pressure idea that survives all of this. A tracheal system exists to deliver a lot of oxygen, fast, on demand. This larva only manages depth by shutting that down. It feeds and grows in shallow water and goes deep to be inert. Surviving the pressure and living at the pressure are different problems, and only the first one has an answer. One more thing worth taking from this animal, and it has nothing to do with the ocean. A wall that changes length when you change its chemistry does mechanical work on a chemical signal, with no motor, no wires, and no moving parts. The lab measured that work, They set the sac's chemistry so it swelled, then used pressure to push it back, and the swelling held against the equivalent of about 80 meters of water. People have been trying to build pH-driven actuators for years. Artificial muscle, valves that open when a fluid's chemistry shifts, pumps small enough to sit on a chip. This is a working one. Nobody has built anything out of it yet. For decades, the reason there are no insects in the open ocean was a fact about insect bodies. It is now a question about salt and timing and who got there first. And the thing that changed the answer was a transparent larva you could lose in a teaspoon of lake water, riding a pair of rubber air tanks down into the dark, every day, on a schedule.